right, hello and welcome everybody. My name is Devin Lau and I'm the Assistant Director here at Yale Center Beijing. A special welcome to all of you guys that are joining us for the very first time. Quick introduction, Yale Center Beijing was established in 2014 as a way to gather leaders from Yale, China, and the rest of the world for dialogue and exchange. We bring the best Yale professors and alums to China to share from all fields of study, from medicine to philosophy to music, economics, et cetera, et cetera. In this time of pandemic, even though we're not able to meet in person, we are very lucky to be able to meet online and to have this platform to reach even more people than before. And so we're very lucky today to have Professor Akiko Iwasaki. I will introduce her momentarily, but I just also wanted to say that today is a very special talk because it's a part of a series we call the Yale Starlight Science Series, where we highlight the best that Yale has to offer in science research in particular. Coincidentally, actually, our last in-person speaker to be featured for this series was Dr. Albert Ko, who spoke in September about the new global threat of emerging infectious diseases. He impressed upon us the very real possibility that there would be more and more outbreaks of novel diseases that would impact the whole world. That was back in September. We had no way of knowing just how relevant and prophetic his words would become. And so from the very last in-person Yale Starlight Science Series talk to today online, we get to hear from Professor Akiko Iwasaki, who is the Waldemar von Zedtwitz Professor of Immunobiology, as well as Molecular, Cellular, and Developmental Biology. After receiving her PhD from the University of Toronto, she joined Yale in 2000. She's won numerous awards, and she's also, you've probably noticed, frequently featured on TV, newspapers, and social media, and has become very popular. She was already very, very popular before, but has been especially popular ever since COVID-19. Her research has resulted in paradigm shifts in the understanding of immune response to pathogens, as well as in vaccine design. I'll let her get into the specifics of the science, but I will point out that she has quite the team of PhD students and postdocs working under her, and her research covers everything from the common cold to STIs to even more, headline drawing diseases like Zika and SARS-CoV-2. And in general, her laboratory is interested in recognition of viral infections and how that generates adaptive immunity within the body, and thus how that can eventually lead to protection against subsequent challenges from viruses. So in general, I just want to say we are very lucky to have Professor Iwasaki, and she's a tremendously popular professor, a noted researcher, and of course now her work is more relevant than ever and very much on everybody's mind. And so we want to welcome her. I would like to ask that by welcoming her, if people are willing to turn on their cameras, that's always helpful, just so that professor is not talking to a screen of black and just, it's always nice to be able to see who we're talking to. So for those of you guys that are able to, it would be great if you could turn on your video cameras. And so with that, I will turn it over to Professor Akiko Iwasaki.
Thank you, Devin, so much for the invitation and your very kind introduction. I'm going to share my screen now. Okay. Can you see?
Yes,
okay.
that looks great.
Great, excellent. So yes, so I'm going to spend the next few minutes with you talking about our work on trying to understand immune response to SARS-CoV-2. And I want to begin with a little introduction of this virus before diving into the immunology. So SARS-CoV-2, as you probably heard already, is a member of the coronavirus. It has a very large genome of about 30 kilobase, and it's a positive sense RNA virus. It's enveloped, and let's see, as you've heard, the spike protein, which protrudes from the envelope, is the target of antibody as well as a lot of vaccine strategies. However, there are many other proteins and components, in particular the viral RNA, that is encapsulated in the nucleocapsid protein that distinguishes this virus. And there have been many other coronaviruses that have circulated in humans prior. And some of these coronaviruses cause common cold viruses, such as the alpha coronaviruses and beta coronaviruses. However, there have been also other more lethal and pathogenic viruses that are zoonotic coronaviruses that have emerged in 2002, as well as the MERS-CoV that has a pretty high lethality. And this SARS-CoV-2 hijacks the cellular machinery to generate more copies of itself. And the ACE2 is the receptor that the virus binds to in order to enter the host. And once that happens, the virus can inject its RNA. And the RNA can be used to make more viral proteins, as well as to make more genome, and eventually be packaged to make many more of them, the copies, to be transmitted. So before the pandemic, what did we know about this type of coronaviruses? So it's been known that antibodies to the common cold coronaviruses do not last for very long time, and that's been sort of a concern that people have discussed about the coronaviruses, and people have antibodies to the common cold coronaviruses do get reinfected, but they're mostly protected from any symptoms. So even though they can replicate the virus to some extent in their nasal cavity or respiratory tract, it usually doesn't lead to much of a disease. So this is, it's good to remember that even the waning antibody levels that you find with serology tests for SARS-CoV-2 doesn't necessarily indicate that you're no longer immune to the virus. And then the long-lasting antibodies do develop in people who have recovered from SARS and MERS. Vaccines against coronaviruses are obviously being made right now. It has not been licensed yet for most of the parts of the world, but this effort is going to lead to hopefully long-lasting and very protective immunity. Another thing I want to mention is that we've also been studying influenza viruses and rhinoviruses. And there, what we've discovered is that not only is the host immune response important, but the host immune response is also under the influence of the environmental factors, such as the temperature and the humidity. And this becomes, I think, very important during the winter months, where the humidity drops as we import dry air into the homes to be heated, and also the temperature outside drops. So this combination, we suspect, is what's leading to the seasonality of other respiratory viral infections, such as the rhinovirus and the influenza viruses. And so whether that also applies to the SARS-CoV-2 is still being investigated. However, what we discovered is that there is an impact of these temperature and humidity on the host, regardless of what the virus is. So it's probably very important to keep in mind to regulate the indoor environment in order to best maintain your respiratory immunity. So what needs to be done to overcome this pandemic? Well, me being an immunologist, of course, we want to understand the immune response to COVID-19. And I think that holds a key for many things, such as whether there are immune responses that are particularly suited to recovering from disease versus those that drive the pathogenic disease. And also, this will inform the kind of vaccines that's needed to protect the host from ever becoming infected. There are also development of antiviral agents that's important, not only for this virus, but going forward for other pandemic viruses that will emerge in the future. Of course, safe and effective monoclonal antibodies and vaccines are going to be absolutely important. as well as drugs to promote disease tolerance. Now, this disease tolerance concept is to basically tolerate the presence of pathogen without having to clear the pathogen. So, for instance, if the immune response to the virus is making us sick, can we stop that immune response rather than tackling the virus? And so these are the types of things that are being tried right now. So we have to kind of basically look at all angles, try to stop the spread of the virus as well as to mitigate the effect of the virus in our host. So since late February of this year, my lab has kind of shifted focus from all the other investigation that Devin mentioned that we are doing in the lab to really studying COVID-19. And so today I'm going to talk about mostly this human work that we've done looking at immune response in the patients. However, we've also created a mouse model of COVID-19, which is very versatile and useful that hopefully people can use to investigate their own within their own laboratories. And also understanding neuroinvasion and neuroinflammatory potential of this virus, as well as the impact of maternal infection in the pregnancy outcome. So, but today I'm going to focus on the first two topics, which is the immune responses that occur in patients and how that dictates host recovery versus disease. So I'm going to jump right into this topic. And so Devin mentioned, it's interesting, the last in-person seminar of this series was given by Dr. Albert Ko, who is my wonderful ally and collaborator, who basically allowed us to engage in this research that I'm going to discuss today. So Dr. Albert Ko, as being an expert in many different pandemic settings, was very quick and effective in gathering collaborators such as myself to come together early in this pandemic to start this respiratory infection by repository program, where we have set up 3 different kind of collection cohorts. The first cohort is the inpatient cohort, in which we have enrolled patients that are coming to the Yale New Haven Hospital to collect their nasopharyngeal and oropharyngeal swab, as well as saliva, urine, feces, and other biomaterials. And we've done this in every 3 to 4 days during their hospital stay. And that had allowed us to really look at the time course, the longitudinal nature of the immune responses, as well as viral replication in the patients. And we also have set up a second cohort of outpatients in which we will also collect various biospecimen to monitor what's happening in milder diseases. And then we also have the healthcare workers cohort, in which healthcare workers working at Yale were recruited into the study, not only to sort of serve as a healthy control, but also to monitor their viral load so that we can, as quickly as possible, detect any exposure and infection that's occurring in the healthcare workforce. And that allowed us to pick up a few healthcare workers that were infected much earlier than they would have if it wasn't for this biorepository program. So what we did in my lab is to collect the peripheral blood mononuclear cells from the patients, and then done several different types of assays, including flow cytometry, which allows us to look at the proportion of leukocytes and lymphocytes in the blood, as well as the status of T cell activation and cytokines that are produced and are released into the serum. And this allowed us to look at a global picture of the immune responses that are ongoing in patients over time. And we've divided the cohorts into 6 different clinical scores. The healthy volunteers are labeled 0 because they obviously are healthy and have no clinical scores. However, the ones that are hospitalized, we divided them into those that require no supplemental oxygen, all the way to having to require mechanical ventilation in the ICU. So by sort of looking at these patients that fall into these various clinical scores, we were able to study the correlation between the immune response and the severity of disease. So the first thing we noticed, as have been reported by many other groups, is that there is a decline in number of T cells, the T cell lymphopenia that happen in patients. And inversely, we see increase in monocytes and granulocytes, such as neutrophils and eosinophils, that come up in the patients. And this is especially more severe and more pronounced in the severe cases. So here, the coding is the healthcare workers are in green, moderate patients are in blue, and severe patients in pink. And we've also looked at over 70 different cytokines and growth factors. And what we see is that there's an elevation of certain cytokines, such as IL-6, which has also been reported, as well as interferon and many chemokines and other effectors, cytokines. And for instance, IL-1 beta and IL-18 are products of what's known as the inflammasome complex. And so these types of very severe inflammation is occurring in these patients. And a clue as to why some patients become severe in disease versus those that are not came from this longitudinal analysis of viral load in the nasopharynx. And what we see is that even though at any given time post-symptom onset, there's not a huge difference between these moderate versus severe groups. What we find is that the severe patients tend to maintain high levels of virus in their nose, and whereas the moderate patients are able to clear the virus ultimately. And this is probably leading to the prolonged inflammatory responses in the severe patients, whereas the moderate patients are able to regulate this ultimately. And interferon and cytokines also were correlated within the patients. So for instance, this is looking at the viral load in the x-axis, and in the y-axis, we're looking at various cytokine levels, such as type 1 interferon, type 2 interferon, TNF, and TRAIL. And what we see is that regardless of the severity of infection, there is a strong correlation between the viral load and these types of antiviral interferon, suggesting that the interferons, even if they're secreted to high levels, are not able to clear the virus. Not only that, when we look at the levels of these cytokines in patients within the first 12 days since symptom onset and ask what are the levels in those patients that end up dying from infection, which is these groups, versus those patients that are discharged to their homes, what we see is that there's higher levels of these interferons, IL-1 receptor antagonists, and many other cytokines in the patients that ultimately died of infection, meaning that the early onset of these cytokines and interferons are correlated with mortality. And we've also seen the same pattern when we looked at the duration of hospital stay. So, it correlates with mortality as well as the length of hospital stay for these patients. And what's interesting when you look over time for various different cell types and cytokines is that, if you just pay attention to the right side of the panels for each of these, you see that in the severe patients, there's an elevation of these cell types as well as cytokines over time, whereas in the case of the moderates, they do have either a slight increase, but sometimes maintaining the level, flatline or reduced level over time of these types of cytokines. And the way we divided these factors is that normally the immune system fights infection using different effector functions. So for instance, the type 1 immunity is important for viral infection. Type 2 immunity is really used for fighting worm infections, helminths. And type 3 is really dedicated for antifungal response. And yet we see elevation in all of these types of immune response, indicating that there's some kind of disorientation or confusion within the immune system that's kind of firing everything they have in the severe cases. So this was a big surprise to us. So we asked the, sort of algorithm to come up with clusters of patients based on the levels of these cytokines in an unsupervised manner. And what we find is that there are 3 clusters of patients that come up based on the cytokines that are, based on 4 different signatures of cytokines. And cluster 1 has an enrichment in these tissue repair growth factors, whereas cluster 2 has enrichment in further the chemokines, as well as these mixed signatures of type 1, 2, and 3. Whereas cluster 3 has this really, really intense type 2, 3, and mixed cytokine signatures we see. And so what do these clusters mean clinically? When you look at the age distribution of patients that belong to these clusters 1, 2, and 3, there really isn't that much of a difference, except that perhaps there's a little bit elevated or older age group in 2 and 3, and then length of hospital stay is not dramatically different. However, what's very different is the disease trajectory of these patients. So in cluster 1, the patients do a little worse, but then they ultimately recover from the infection and they're discharged. Whereas cluster 2 and 3 patients, they develop a quite rapid decline in disease, meaning that they have higher clinical scores, and ultimately some of them end up dying if you look at the mortality scores here, as well as slightly increased coagulopathy that are seen in cluster 3. So this indicated that, but based on the cytokine levels alone, we're able to kind of predict what kinds of patients, how the patients will ultimately do with their disease, and that the growth factors that repair the tissue damage appears to be enriched in the ones that do well with this disease. And then so we asked for biomarkers for COVID-19 mortality based on all of the data that we have collected. And so just to orient you, the lower the score, the better prognosis in terms of mortality these patients will encounter. And 1 is neutral, and 10 is very high correlation with mortality. And what we're seeing is IL-18, which I mentioned is a product of the inflammasome activation, is one of the highest mortality factor for this disease, followed by interferon alpha, IL-10, and so on. So this led us to kind of, gain a much clearer picture of what kind of inflammatory responses are most associated with mortality. So looking at this first longitudinal study, what we found is that the persistent viral load is detected from severe disease patients. And this viral load appears to be driving the inflammatory responses. And increase in these types of cytokines and interferons within the first 12 days of disease is actually correlated with mortality and hospital length stay. As well as these, we've uncovered 4 different signatures of genes that correlate with patients who do well from this disease versus who progress to worse disease. And finally, there appears to be these misfiring of cytokines that belong to many different kinds of immune response being elevated in the severe cases. So what does it mean? When I teach my medical students about immune response, we often, start to talk about interferon being a very important antiviral response. And it's true that if you develop very good interferon response, you can control the viral load and recover from a viral infection. Whereas if you don't have a rapid interferon response, you end up having more viral load and then having a persistent level of interferon later on is usually associated with worse disease outcome if you look at other coronavirus infections. So our data appear to point towards this latter possibility, that there may be this prolonged interferon response going on in the patients that is associated with worse disease outcome. So now I'm going to talk about using this data set to probe some of the host intrinsic differences in immune responses. And here I'm going to focus on sex difference in immune response to COVID-19. So as you all know, the male sex is a risk factor for severe disease with COVID-19. And this has borne out from many different countries, including US, China, as well as Italy, and many other places in UK. So we wanted to know whether we can use this data set that we've generated to probe the importance of sex in determining the kinds of immune response. So the way we designed the study is to look at baseline analysis of the immune response in the patients, meaning that we are only going to look at the first time collection from patients who are not in ICU, and who have not received any immune modulatory drugs, such as tocilizumab, which is an antibody against IL-6 receptor, or high-dose corticosteroids. And the reason we focused on this baseline analysis is that we wanted to understand the sort of natural state of the immune response in patients before they receive these types of immune modulatory agent in order to understand sex differences as opposed to drug-induced differences. We also, so in this patient cohort, the baseline cohort, which I call cohort A, has 39 patients, 17 men and 22 women. We also recruited healthcare workers, and then we also extended the analysis ultimately to a larger cohort where we look at the parameters that are predicted from the baseline analysis to 98 patients. So here, first, what we found is that looking at the viral load in the nasopharyngeal swab or the saliva, the male patients, which are in orange here, versus female patients, were not significantly different with respect to the viral load, even though you can see that the mean viral load appears to be higher in the male compared to female. But the distribution was so wide that it was not significantly different. And similarly, when you look at the anti-spike IgG or IgM in patients, there were no statistically significant difference between female and male. And we also found that many factors that we've looked at were elevated in the patients regardless of sex. So for instance, CCL5 is quite elevated in both sexes, and CXCL10 is highly induced in both sexes. So a lot of the cytokines fall into this category. However, there were a few cytokines and chemokines that were more elevated in male patients, such as IL-8, which is a chemokine that attracts neutrophils, is highly elevated in the male patients compared to the female. And IL-18, again, comes up, this inflammasome-mediated cytokines are elevated in male versus female. And even in terms of the cell types that are enriched in male versus female, we also found that this inflammatory monocytes were elevated in male compared to female. And these inflammatory monocytes, are recruited to the lung based on different chemokines such as CCL5. And we see this correlation between the level of CCL5 and inflammatory monocytes that are in circulation in only male, but not in female. So there seems to be a male-specific elevation of monocytes based on the CCL5 levels in these patients. Conversely, though, when we looked at T cells, which are the sort of key adaptive immune cell types that are important to fighting this infection, we saw elevated activation of T cells in the female compared to the male patients, especially of the CD8 T cell kind, which are the sort of the killer cells that detect infected cells and eliminate them from the body. So we've learned some differences between the sexes at baseline, but what can this data tell us about the disease trajectory? So then we divided the male and female patients into two groups. Those who did poorly during the course of study, which are in this disease deteriorated group indicated by filled circles, and those that maintain stable disease, so stability of scores 1 and 2. And again, we divide them into male versus female. What we saw is that in males only, the ones that did poorly with disease have older age, whereas in female, there were no difference between those who did poorly versus those who had stable disease. Similarly, the body mass index was elevated only in male patients who did poorly with disease, but not in female. And I told you that male patients tended to have lower level of T cell activation. But among those, if male patients made very low T cell activation, basically this is a baseline level of T cell activation, they also belong to the disease deteriorated group, whereas the ones that were able to elevate somewhat the T cell activation, they were able to maintain their disease. So their correlation is pretty striking that males that developed very low T cell activation did poorly. Whereas female, there was no significant difference between these. And in fact, if you look at the interferon gamma secretion from CD8 T cells, again, a very important antiviral cytokine, they were reduced in the ones that did poorly. And conversely, female patients, even though they're able to make T cells, activate T cells, if they made large amounts of these innate chemokines and cytokines, they also tended to do worse with disease. So there seems to be this sexual dichotomy between the adaptive and innate in predicting how they do in their disease. So this is a striking figure, I think, because what it shows you is that the age, the relationship between the older age and the T cell activation status is inversely correlated only in men. See, this is looking at the orange here again. The older the men become, the less activation of T cells and less interferon gamma from T cells are seen, whereas women, even when they age, they actually seem to maintain very well the ability to activate T cells. So that may explain some of the differences that we've seen so far. So to conclude my talk, basically men were found to develop more inflammatory cytokine response than women at baseline, whereas women were able to activate T cells better than men. And men who developed very low T cell activation belonged to the deteriorated disease group, whereas women who developed inflammatory cytokines did worse with this disease. And finally, women age better with respect to the T cell immune response than men. So I want to end here by thanking my entire lab members who have been really working around the clock trying to understand the immune response to this virus, as well as implementing testing in the Yale New Haven community in order to help out with this pandemic. I also want to thank again the Albert Koh and others who are in this impact study team. This is the biorepository team that we've set up together. And there are a number of other investigators, Saad Omer from Yale Institute for Global Health, Charles de la Cruz, Shelley Farhadian, who are clinicians who are on the front line treating and consenting these patients. Nathan Grubal, he's an amazing molecular virologist who are studying viral evolution and viral mutations in these patients. Craig Weiland, a great colleague who is in the BSL-3 facility all the time trying to understand the virology of this infection, and Aaron Ring, who is a great collaborator who did all of the ELISA assays for from the patients, and some funding that allowed us to carry out this research. And I'm just going to end with this slide showing my amazing lab members who really selflessly dedicated themselves to working on COVID, and without whom I won't be here today to tell you about these stories. So thank you for your attention.
Thank you. That was amazing.
Thank you.
Learned so much. Great. So, and also you were able to explain everything so succinctly that we have a good amount of time for questions and answer. So if people have questions, you can either type them into the chat box on the bottom of your Zoom screen, or you can directly raise your hand under the participants section, and I can unmute you and you can talk directly to the professor. Okay, so Howard has a rather specific question.
Yeah, that's an interesting question. So I'm just going to repeat it for the people. Yes. So Howard asked, do we see less severe response in prostate cancer patients who are under chemical castration? So I think what Howard is referring to is the role of sex hormones in dictating immune response to the COVID-19. And so we didn't specifically look at sex hormones yet. That's something that we're currently actively looking at. And we also didn't have enough patient number to really make any conclusions about whether prostate cancer patient, I don't think we even had any in that cohort. So that's something that we would like to measure by measuring sex hormone levels in all of the patients. So that's what we're doing right now.
And because we have a good amount of time, I'll probably, even when people type out the questions, I'll probably unmute you guys so you guys can still interact a little bit or explain what you're thinking by asking your questions. So I guess while people are still thinking of questions or typing them out, I guess one question that I had was, in terms of the differences that we see in sex and response, how typical would you say this variance is compared to, say, other coronaviruses or other viruses in general? Is this sort of a typical response that we see?
Right. So unfortunately, we don't really have this kind of in-depth analysis longitudinal for other viral infections to compare to. So we can only kind of compare, snapshot of what happens in other coronaviruses or other viruses. And what we notice, obviously with SARS and MERS, you do see the cytokine storm, and you do see a lot of similar clinical features. And this eosinophils and this sort of type 2 immune responses have also been reported for especially older hosts. So it's possible that the reason that the very severe diseases are occurring in the older cohort has to do with their propensity for immune dysregulation. And we've also shown in previous studies that the interferon response is really blunted in people over age 65. And so we compared in that study, 65 to 90-year-old versus 20 to 30-year-olds in the ability of their monocytes to secrete interferon. And there was a huge difference there. So that could also contribute to this, uncontrolled viral load that we're seeing in older patients. Okay, so.
It's like Agnes has a question regarding research about how long antibodies last after recovery. And I know you just came out with a study about whether that was, or you know, in the article about whether that's even a accurate thing to say, or how we need to specify what we mean by that, right?
Absolutely. So yeah, this is a huge topic of interest in public, and that's why I kind of included that in the introduction. So antibody after recovery, multiple papers have come out that says that, you know, within 2 to 3 months of recovery, the antibody levels are waning, meaning that they're dropping, and that you know, has caused a lot of concerns about whether we're ever going to have vaccines, and that's a very different question. Whether a vaccine can elicit a long-lasting antibody response is very different from whether a natural infection can do it. And the reason for this is that vaccines can be designed against a specific antigen with the inclusion of adjuvants. And adjuvants are what stimulates the innate immune response that triggers, you know, very robust active B cell immunity, as well as T cell immunity that allows for long lasting and robust antibody response. So I'm hopeful that among the 150 vaccines that are being currently tested, that many of them will end up doing much better than natural infection for antibody response.
All right. I see that people are warming up and the questions are starting to roll in. So Jenny, I'm going to unmute you so you can ask your question directly.
Hi, my question is about the relationship between a blood type and susceptibility to COVID-19. I heard that type A is particularly vulnerable. Is that true?
Right. So I have also seen those reports of blood types being correlated. And that's a very interesting area of study. But you know, the relative risk for developing more severe disease with the blood type is very modest. So it's not like if you have blood type A, you're just doomed with this infection. Not at all. This may be telling us something about the relationship between the blood antigen and the immune response to this virus. And that's something very interesting that we and others are investigating. But yeah, there appears to be some correlation with blood types.
Right, Howard, I'm going to unmute you because you have such great questions. If you don't mind asking directly.
Hi, Dr. Iwasaki. I'm a fan of you in the Twitter world. It's nice to see you in person.
Nice to see you.
So my question is, have you seen any outliers in your patient that's specifically correlated with any specific type of autoimmune disease, which is like, you know, if you have a strong autoimmunity in one particular immunity, whether you get severe or better outcome?
Yeah, so again, our study currently isn't powered enough to really look at those issues yet. We only had a handful of autoimmune patients in our cohort, but there are people who are looking at this at a much larger scale, you know, tens of thousands of patients. And so I think there we will learn a lot more about whether autoimmunity will actually predispose somebody to worse disease. And of course, autoimmune patients are also taking, you know, different immune modulatory drugs, right? So that will also confound this kind of investigation. But yeah, so autoimmunity, cancer, any sort of type of existing conditions that would modulate the immune response, whether that contributes to worse disease, is an area of active investigation.
Okay, thank you.
Thank you.
All right, Tim, I'm going to unmute you so you can ask your question.
Okay, so I'm still a junior in high school, so I'm pretty immature in this field of study. So, but like on some online medical journals, I've seen that like there's RNA vaccines that will be invented there, or like they have the potential to produce immunity in humans. And I'm just curious about like how RNA vaccines can work against the COVID-19 and like the general mechanism of the vaccine.
Right. So, yeah, that's the interesting thing that's happening in this pandemic is that all these sort of unconventional methods for vaccinating are coming to the forefront because of their, speed at which the vaccine can be generated from nucleic acids. So, yes, the mRNA vaccine as well as DNA vaccine, these are, one of the sort of top vaccines that are being tried right now. So the way in which this nucleic acid vaccine works is to rely on the host cells to produce the proteins, the antigens that the immune system can react to. So for instance, most of these vaccines are targeting the spike protein. So the gene that encodes the spike protein is being introduced into the host cell, and that's that's producing the proteins for the immune system to recognize and mount immune response. So we're kind of like letting the host make the vaccine, in-house within the host. And that works well. I mean, I've seen data from clinical trial phase 1, 2, as well as primate studies, and they seem to induce very robust immune response antibodies and T-cell immunity. And so if you want to dig down a little bit deeper and ask why are these nucleic acid vaccines able to do that? Because in order to derive a good antibody response, as I just mentioned, you need adjuvant. And so the nucleic acids themselves are the adjuvants in this case. So what my lab and others have reported is that these vaccines, the nucleic acids, RNA or DNA, are recognized by toll-like receptors as well as cytosolic sensors of nucleic acids that are dedicated to detecting viral infections. And so the vaccine is mimicking not only the protein of the virus, but also the sort of process of virus infection. And that's maybe why these nucleic acid vaccines are working well to develop an immune response.
Great. Next, we have Eric. Eric, if you don't mind asking your question in a more general way, that would be great. Oh, it looks like your sound isn't connected. You're unmuted, but I think he's probably trying to sign back in here. Well, okay. Well, while Eric is trying to get back on, let's move on to the next question here. Let's see. Peng. Go ahead, Peng.
Hello. As my question mentioned, in one development cell, single cell study have mentioned that in the smoking patient, the COVID-19 receptor ACE2 mRNA level is increased, but from the hospital, the statistical analysis shows that the smoker rate among the patient is relatively lower. So I'm wondering whether the smoking cigarette promote or inhibit the COVID-19 infection and disease severity. Thanks.
Yeah, thank you. So yeah, that's been confusing because in the very early stage of COVID, smoking was thought to increase the risk, and then it may not be. So, and whether the ACE2 expression induced by smoking alters the host susceptibility or not. So this is an area that my lab, in collaboration with Dr. Charles de la Cruz's lab, is currently investigating by looking specifically at smokers, either active smokers or previous history of smoking versus those who don't smoke, to really dissect the immune response. So my lab, we're not epidemiologists, we're immunologists. So we're looking at the immune differences. And that's something that hopefully I can let you know in a few months once we analyze these patients.
Oh, thank you.
Thank you.
All right, let's see if Eric's audio works.
Hello, can you hear
There we
me?
Hi.
go.
Yes, we can.
Hi. Thank you, Professor, for your for sharing all this information. I've been taking notes. Thank you very much. I'm sorry for not raising a specifically very scientific question. I think a more general question would be probably as students, rising seniors, how long will it be before we see a working vaccine that would be publicly distributed and working? And if that is not available in a very short period of time, should students like us consider taking gap years or be very, very careful about crowd gatherings offline? Thank you.
Yeah, Eric, that is the sort of biggest topic of discussion at Yale as well. Because, you know, we obviously want the students to have a in-person experience because college is not just about learning, but also about interacting with other students and professors and so on. So of course we understand the absolute need for that, but also at the same time, as you mentioned, without the vaccines, it's going to be very difficult to implement that. So I know that the plan is, you know, to screen incoming students for Yale at least for SARS-CoV-2 by PCR, and to obviously quarantine those who are positive and do measurements on a very regular basis in order to kind of counter this danger of being exposed and infected. But I agree with you, like ultimately, in order for us to go back to what resembles, you know, society prior to pandemic is vaccines. And but as I mentioned, I mean, there are like, you know, 150 different vaccines that are being tested right now, with many of them having entered clinical phases. And what I'm seeing so far is pretty encouraging. You know, titers of antibodies generated much higher than natural infection, and T-cell activation occurring in the vaccinated people. So I'm hopeful. I can't give you a timeline of when this is going to become available, but I'm hopeful that many of these things will work. And you know, it's not about finding one vaccine winner. It's about finding multiple vaccine winners that can be distributed throughout the world. Because we need billions of doses. And for that, we can't rely on one platform. So that's why I'm hopeful for all of these endeavors to come up with something soon. But yeah.
That's actually pretty encouraging. And, there's hope. Great. Let's see, Xiaohui.
We can't hear you, Xiaohui.
Can't hear you.
Oh, sorry.
There you go. Yep.
Yeah, thank you. So thank you for giving us this educational sharing. And we will see your first study as soon as your article gets published days before. And my classmates were thinking about the clinical significance of your study. So since your study mentioned that you already found that there is some correlation, you can subset the patients according to their cytokines or chemokines, like these immunological profiles. So we're wondering what can it be used in clinical context, because we see that this is not for prognosis, since it is measured after the disease. Yeah.
Right. So that is our goal, is to hopefully inform the clinical treatment that is appropriate for the COVID patients. So as I mentioned, there are a couple of times I mentioned inflammasome activation being one of the most correlated with mortality. And there are inflammasome inhibitors and cytokine antibodies that can block the inflammasome mediated cytokines or itself. the inflammasome itself. So that's something that I've been kind of thinking that would be an appropriate treatment for before people become severe for this COVID, especially if the inhibitors are not toxic itself. So, and the reason I say that is because I think inflammasomes, you know, at least from our influenza studies, inflammasomes don't have a major role in controlling the virus infection. But when it's triggered, especially in low settings of low interferons, inflammasomes can become activated. And that led to lethality in the case of influenza. So, you know, learning from that lesson, I think inhibiting inflammasomes is a potential, you know, clinical treatment for patients. Now, you know, any kind of treatment needs to be tested first in randomized placebo-controlled clinical trial. And I know that there are several that's ongoing right now. So I'm looking forward to those results.
Okay, thank you.
Thank you.
Next we have Sean.
All right. Can you hear me?
Yep.
Yes.
Oh, okay, great. Hi, Professor Iwasaki. Sean here from Chicago. So that's why you kind of see the sun in my eye here, an hour behind you. My question is a simple one. Is there any correlation between the patient weight and their disease outcome, their severity of the disease? Perhaps more specifically, is obesity a main factor in disease severity? Thank you.
Yes, thank you, Sean. Yes, we did look at the importance of the obesity using the body mass index ratio. And what we saw was that in extreme high BMI, like morbidly obese or, you know, very obese patients were included in the high risk group, as well as when we looked at the sex difference in immune response and how, you know, their disease progression, especially in male patients, we saw higher BMI correlating with worse disease. So there may even be sex differences between whether obesity leads to severe disease. And that's also very interesting. Like, what is the interaction of sex and obesity? And so that's actually our next investigation using the data set that we have, is looking at BMI and the severity and immune response.
Thank you.
Thank you.
Great. Next we have Lee Yong Chao.
Hi, Professor. I'm Bruce. Thanks for your sharing. And
Thank you.
First, please forgive me as I can't open my video, and I'm on a car in China where it's night now. Yeah. My question is, as you have shown just right now, the COVID-19 infection could trigger T cell count drop down in patients' plasma, and the CD4 positive T cells count is correlated with better prognosis in patients. So my question is whether the behavior of this COVID HIV virus. If the answer is yes, what can we learn from the strategy we used to treat or prevent the HIV infections? Thank you.
I'm sorry, some of the portions were cut out and
Right.
it was.
So I think the gist of the question was, given the effect of SARS-CoV-2 on T cells and how that has been affected, is there any correlation or similarity to, for example, HIV, which has a lot of, you know, T cell interruption? And in that case,
just show two results about COVID-19 infections can drop down the T cell count in.
Yes.
Right.
I got it. I got your question. Yes.
patient's plasma. And
Okay, maybe I can answer that question. So yeah, that's right. So the T cell numbers decline quite rapidly in COVID patients, and that may contribute to, you know, why it's difficult to deal with this virus, because, you know, T cells are essential for fighting viral infection, and even to generate antibody response, T help is essential. So when we're losing the key master regulator of the immune response early during this infection, the patients do have somewhat of an immune compromised state. And that may be leading to the ability of the virus to replicate so well. But in people who have a very mild or asymptomatic infections, whether this kind of T cell drop is also happening or not, that's an area of investigation I think we need to move into, because what we've studied so far is this very severe cases, and when we call moderate cases, these patients are, you know, still in the hospital and having to receive oxygen. So whether that T cell lymphopenia is really the culprit for severity of disease, we need to look at asymptomatic people.
Next question comes from JZSH, but I think this person dropped out. But the question was whether, with the study that you have on cytokines, I guess, what is the general process of a study like that, and then taking it to the process of some kind of clinical outcome? So I guess the general question is, you know, in such a process, how long does it usually take? And I guess, so that I can ask a more general question here, the amount of studies that we're seeing and the, you know, time and energy invested into the studies, you know, how unprecedented is this kind of effort to do all of this as quickly as possible?
Yeah, so it is, I mean, so to be honest with you all, this is my first time doing this as well. So this is my first human translational immunology investigation. So I'm learning as I go along, and yeah, it's heroic amount of work, you know, not by me, but members of my lab as well as collaborators. And what I'm learning from this kind of investigation is that you really need enough patients. You know, if we had half the number of patients, we would have drawn wrong conclusions. So first, you need enough number of patients. You need also, in our case, we collected patient sample every 4 days. And that was really important, because without that, we couldn't have drawn the conclusion about disease trajectory within each patient. So having a dedicated team working, you know, very long hours, unfortunately, collecting enough patient sample from enough time point, provided us with the critical tool to investigate. And I know that my group is not the only one. Obviously, there are many other groups in the world doing this kind of research. And I think I really commend everyone for doing that, because it's really important to understand in real time how the immune system is responding to the virus. And as you see from our study, like, there's a lot of important insights that can inform clinical treatment and potentially inform vaccine efforts as well. So yeah, it is a difficult work, but I think many people are dedicated around the world to doing this. And the other interesting thing is, you know, all these studies that are coming out of other places, we're also trying to compare our results with others. And not all the studies agree on every point, right? But that's interesting, because that means that there's something different about the patients, or maybe the way the analysis was done, that would, you know, come up with different conclusions or different results. And that's also a learning opportunity. So we're looking at, you know, all of these data as they come out with that vision in mind.
All right, next we have Rain.
Hi, Professor. Thanks for your sharing. And well, I'm a bit confused about like the antiviral agent you mentioned earlier. Like how can this kind of agent help the patients to overcome the disease? Because I've heard that well, this agent cannot really cure the disease, but it can like inhibit the replication of the virus.
That's right. So right now we don't have a specific antiviral for the SARS-CoV-2. A lot of the antivirals like remdesivir and so on are being repurposed from other viruses. So what I meant by need for research is to develop specific antivirals for different classes of viruses. So for instance, even though coronaviruses differ in many different sequences, a lot of the kind of replication complex and so on, there's a similar mechanism and similar target that we can make drugs to inhibit. And so, you know, if we, let's say if we had a equivalent of antibiotics, right, for all viruses, imagine the kind of response we have, right? So even for flu, we have, you know, several antivirals that people can take within the first few days of infection, which prevents this very worst disease outcome. So that's what I meant by needing antivirals, because if we have a shelf full of antivirals for every kind of virus, the way we can approach the pandemics in future will be very different.
Great. Next we have Celine.
Hi, Professor. Thank you for sharing. My question may be similar to the one before, because I'm very interested in public health, but I know little about medicine. So I was wondering, your study on the immunity of this virus and the other groups. I mean, on the studies on this disease. How would they have the influence or impact on the development of HIV vaccine and other infectious disease? Yeah, because for me, I think they are kind of related. Thank you.
Mm-hmm. Thank you, Celine. Yeah, so I've been very interested in developing vaccines against many different kinds of viruses. And each virus poses different challenge. HIV, for instance, as you know, is highly capable of mutating even within the host, with, you know, accumulating mutations to evade antiviral immunity. And that is why it's very difficult to design a vaccine against HIV. Whereas coronaviruses, you know, don't have that kind of mutation capability. And I feel that it's much easier to make a vaccine against the SARS-CoV than to make a vaccine against HIV. And so, you know, in terms of the evasion mechanism, it's much more easy to handle than HIV or even herpes simplex viruses, which I've been studying for 20 years. It's very difficult to make vaccines because herpes has a glycoprotein that inhibit the ability of antibody to act on them. So, you know, that makes it very challenging, whereas coronaviruses don't have that kind of mechanism. So that's why I think it would be not too challenging to develop successful vaccine against this virus. But we may not be so lucky. The next pandemic may be much more difficult to make a vaccine. That's why I think we need a whole different approach system where we have vaccine candidates, we have antivirals ready to go, we have monoclonal antibody production capacity to develop against, you know, whatever surface protein that the next virus is going to have, and obviously public health, as you mentioned. So all of these have to be ready, you know, to counter the next pandemic, and I don't think we're there yet.
I mean, it's crazy to think that as difficult as this situation has been, this is probably a warm-up for, you know, the big one, right?
That's exactly right. I'm afraid so, yeah.
All right, next we have Jenny.
Hi, Professor. I read somewhere that radiotherapy can be used for patients with severe COVID-19 symptoms. I wonder how that is possible, because doesn't radiotherapy destroy the immune system?
Yeah, Jenny, I also read the same. And I was also wondering about that. But I think what they're trying to do is to kind of suppress the overt immune response that is causing the disease. And in which case, the low dose, I mean, obviously, you don't want to irradiate people with high dose, but low dose radiotherapy might work to just suppress enough of the immune response. But it is also, so that's what I was talking about, this disease tolerance mechanisms. How do we suppress an overt immune response to help patients with severe COVID? And dexamethasone and perhaps radiotherapy and anti-immune or immune modulatory agent are being considered. But they have to be done very carefully, because you could actually backfire, right? Because, if you suppress the immune response too much, then you're going to have more virus replicating, and that may cause even severe disease. So yeah, it's a very difficult challenge to do it with radiotherapy, I think.
And then we have Wendy.
Hello. Could you hear me?
Just very slightly, yeah.
Oh, hello,
if you can speak up.
hello.
Yeah.
Oh, hi, Professor. Thank you for sharing. I want to know, will be the patients totally okay through the vaccine? Will there be a recurrence? Thank you.
It was very hard for me to hear your question, but
I think the question was basically with vaccines. And again, we don't know what kind of vaccine we're going to be dealing with, and there are all sorts of different possibilities, but sort of the issue of recurrence and how effective these vaccines will be. And I guess if we compare it to the typical seasonal flu and colds, and that's only about a year or so.
Oh, I see what you mean.
The issue
Yeah, so, as I mentioned, the vaccines, if they are very potent, should be able to provide protection for years and not just 1 year. However, we don't know. Like, if the vaccine is only effective in producing immunity for a year, then you would have to have booster shots every year. Which I don't think it's going to be the case for coronavirus. The reason we need this for the flu shot is because, well, not only the antibody doesn't last very long with that shot, but also the flu itself mutates every year. So we need new antigen every year. Whereas with this coronavirus, I'm really not sure if such mutations will accumulate every year. So again, I think the challenge is not so dire for making vaccines. And the recurrence, obviously, that's something that we're only going to learn in phase 3 and phase 4 trials, whether a vaccine can really prevent, recurrence. But hopefully we'll learn that over the next couple of years.
All right, and we'll go back to Leon Chao for the last question.
Hi, Professor. As I talked before, and the COVID-19 and the HIV virus has similar behaviors. So could we learn from the HIV therapy as the cocktail therapy used in HIV infections has been reported by a study just a few days ago?
So I think the question was, again, talking about similarities that we've seen in certain aspects with HIV and how cocktail therapy has been sort of a way that HIV has been treated over the years. Is that something that is applicable to COVID-19?
Right. So I think that's a great idea. You know, as soon as we develop specific antivirals, they can be combined to make sure that mutations that escape those antivirals don't emerge. And that's the whole purpose of having cocktails. So yeah, I mean, in a ideal world, you would want to have, you know, 2 or 3 antivirals as cocktails to be giving to people. That would be great.
All right, thank you all for a fantastic round of Q&A, and thank you, Professor, for staying and answering all those questions. You're incredibly patient and generous with your time. And also thank you for being able to explain such a complicated and constantly evolving topic in such a easy to understand and direct and hopeful way, really. So thank you very much.
Yeah, thank you all for coming to my seminar, and please stay safe.
Yes, so I'm going to unmute everybody so that they can say thank you to you personally. And then if people are willing, please turn on your camera so that we can take a group picture to celebrate.
Thank you, Professor, for
Professor.
your
Thank you.
questions.
Thank you.
Thank you so much.